And then we'll move on. So officially we would have to move on now with Gooitsen van Dam, who, as already alluded to before, cannot make it, which I really regret. The good thing is that that puts us right back on time. So we'll continue with the FDA view on things. Frank Weichold will be giving the talk, and very much looking forward to hear how you guys think about this. Please.
Thank you very much. Thank the organizer for inviting us, specifically me, actually. Also, Beat Löffler was mindful enough to sandwich me right in the middle between very excellent research data and experimental data, which I will not show you. And as such, as soon as I figure out how to advance this, you need to be aware that is my personal opinion, and I need to state this. How do we think about Theranostics? Well, first of all, please do understand, it's nothing different how FDA approaches that, because for FDA, a name is not a regulatory trigger. Theranostics doesn't change the approach how we regulate product. And we do not regulate technology, we regulate product. A regulatory trigger would be a definition, for example, drug. Drug is an article, and you can find this all online, but let's just, for the sake of the discussion here, remind ourselves about the definition of a drug, something that's recognized in the pharmacopoeia or the formulary. It's also an article that is used for diagnosis, cure, mitigation, treatment, or prevention of disease in man and animals. And articles in food intended to affect the structure or any function of body or man and other animals, such as hormones, for example. It wouldn't be a disease. Pregnancy is not a disease. And any composition thereof. So Theranostics, even though you say, well, there's a diagnostic element to it and a therapeutic element of it, it all could fall under drug. And as such, I will go a little bit more into detail how the Office of Drugs, the Center for Drugs, actually regulate that. We have some legal authorities to do that, you're aware. Food, Drug, Cosmetic Act is the underlying thing, Public Health Service Act, the FDA Amendment Act, the Safety Innovation Act, and of course the User Fee Acts, which provide FDA with authority and also resources to regulate. Now you're all aware of the GMPs, the Federal Code of Regulation, CFRs, and certainly we write guidance documents, some of which I will reference to. Those are usually published and developed in dialogue, so in a way you're part of the dialogue. Now, when you think of combination products, there are some differences, but for devices perhaps a little bit more. But just to illustrate, many of the underlying principles to regulate product are very similar. But we also, of course, need to develop strategies that we call our case law, and as such, the case law has to be consistent. It cannot be arbitrary, and certainly it shouldn't be capricious. As such, we use science as a underpinning for decision-making processes. However, science itself should provide the information, and it cannot make decisions. Decisions are made with judgment. Judgment is put in a context, in the bigger picture, to the benefit of society or a health situation that we have in society. And of course, if you ask different stakeholders, you will get different answers. So it's also true that the less data we have, the less information we have, the less knowledge we have, the more uncertainty, the greater, of course, is the judgment and the question about values. Now, that brings me to another important point. Of course, we have a need for decision-making analysis. We need to analyze how we make decisions. And certainly we want to have a dialogue how we do that. FDA, sometimes referenced as a standard in terms of decision-making and regulation of product, because we use evidence-based medicine to do so. And I think in the past 2 decades at least, the position of the agency towards the dialogue has significantly changed, and we're all part of that. So as I pointed out, we don't regulate processes, and we do not regulate the terminology necessarily. We need the terminology to get on the same page. But in some cases, even though we focus on the product, we have to look into the process because the products are so complex and complicated, and many of you have pointed to some of those very elegant new ideas and build very complex systems. We have to look into how is it made and how it can be made in a consistent way, and how do we characterize that. So in a sense, we are all aware of the diversity, and I don't have to go into much details because many of the speakers with the different presentations have alluded to that. If you build such a complex system, you're challenged. We try to help ourselves and our stakeholders with defining some of our approaches with guidance documents, and some of them are published, some actually 2 are in draft. One very important one from CEDAR in the context of drug, which was supposed to come out early in 2016, but you know how it is sometimes with regulatory agencies and bureaucrats, it all took a little longer. So it's still a draft guidance. Now, if you consider that, let's say, a diagnostic and a therapeutic is not just a drug, but it could be a combination product, in such case the agency resorts to an elegant way. We have an office of combination product that's directly under the office of the commissioner, and this office is specialized in making such judgments as to who is the primary review for this particular product. I list some examples. It's not so important. It's important for you to understand that there is consideration for such primary mode of action when it comes to the regulation of a product. Specifically for CEDAR, the approach is, of course, to take also a risk-based approach. So we did some exercises in terms of risk assessment. Of course, there are technical elements to it, the technical part, but also in terms of what do we use for regulation. Do we have enough regulatory authority? Do we have the right documentation? Do we have the right guidance documents, for example? And to show you a little bit more of the detail, that's, if you're interested, all published. We use, for example, Ishikawa diagrams to look into risk factors. And we also do gap analysis in terms of the challenges that we face up to and what is in place and what is missing. This is just an example to highlight that, in fact, for all the considerations, for example, what is the product, where is it applied to, in which compartment does it end up, how is it metabolized, if it is metabolized, how is it accumulated, and all those kinds of things. Analytical methods play a very critical role, and I think one of the opportunities also of this audience is it to continue the dialogue, to build consensus, to have good communications and consensus standards, which we can use as a platform to interact and measure apples with apples. I think is one of the important elements and opportunities how we can speed up the process of regulation, but also the process of new drug development. So, in a way, you can categorize products into different risk categories: high, medium, and low. I think in a practical way, this is straightforward, and of course, it's always difficult to give an abstract talk when the devil is in the detail. I mean, products are so different. What do you focus in? So, but at least I want to share with you the general approach that the agency is taking. So, for risk assessment, we focus on CMC, biopharmaceutical questions, pharm tox questions, and clinical pharmacology questions as well. Some of the research priorities, because as a regulatory agency we believe that we have to have expertise at the agency in order to regulate product. we need to understand the product. And those are some of the areas that I highlighted here, which are the research focus areas, including the solution testing, in vitro systems for release testing. But again, the devil is in the detail, and not everything behaves similarly, even if you try to develop a reasonable model. And certainly permeabilization, absorption, if there's metabolism, those are very considerations in context of the evaluation. Now, the agency in itself has formed a network within, and I'm not here the first year, so you've probably heard me mention that. We have a working group or a task force that interacts with all the different centers within the agency. Certainly we focus on scientific communication. We train our reviewers. We also have a core facility at the agencies, research facilities that allows us to do physical-chemical analytics and have an influence on the consensus standards. But it is important for you to understand that FDA alone cannot develop standards. That would be somewhat a challenge because industry wouldn't like it, because they would feel that we dictate the methods or the view on things, and as such, to develop meaningful standards, it has to be a dialogue. And my colleague Anil Patri will later on today make some reference to that, but we'll have probably also the opportunity throughout the next 2 days to discuss some of those opportunities. Outside the agency, we interact, of course, with all the stakeholders, and I think that's very important to develop and build this dialogue. Just to remind you, we regulate about 1/4 of all consumer dollars spent in the agency, which amounts to several $1 trillion a year. And we interact through advisory committees. We have over 30. We reach out to specialists and experts. And there's also opportunity for public and stakeholder input in a formal or informal way. And certainly any individual can approach the FDA and make a case. So there are essentially no limitations, but one of the very common ways is to, of course, engage into pre-IND or pre-meetings with the regulator so that the challenges can be discussed. And it's often good to focus on the scientific questions there and not any of the legal framework. In conclusion, I would say FDA doesn't change its general approach to regulation just because there is Theranostic or a new technology. We try to develop a reasonable network within the agency to communicate, to include all experts. In addition to the responsibilities that the centers take and the different review groups, we are building expert working groups that are agency-wide, and those working groups also interact with other agencies as well as internationally. Of course, there are different legal standards among the different product classes, but there's a pre-market review. The attention to nanomaterial is incorporated, where the authority does not provide any pre-market review. There is voluntary submission of data that's mainly in the sector of food. If any product contains material that is not commonly known as low risk, I think it's a good idea to share that with the regulator. And of course, a very important step is, or a very important element in the context is a post-market monitoring. So we are working on guidance documents that are continuously updated. I think it's important for industry to, or any of the stakeholders, to engage early with the agency into a dialogue so that the scientific challenges can be discussed. We continue to train our experts, and we develop what we call regulatory science in nanotechnology, where we dedicate significant resources to be cutting edge. And as I said, international networking and harmonization, and particularly consensus standards, are a critical element to that. So, opportunity is complex. It includes science, law, medicine, policy, but also managerial decisions and execution. And last but not least, you all know, politicians like to get involved, and certainly every stakeholder might have a different angle to the aspect. Importantly, no one has all answers. I think it's important to see the opportunity for collaboration and partnerships, and we encourage all of you to engage actively into this. With that, I'd like to thank my collaborators, specifically Jan Zimak, Celia Cruz, Jed Costanza, Anil Patri, the nanotechnology reviewer networks in the regulating centers, but also the overarching working group within the agency, which is called the Nanotechnology Task Force. With that, thank you very much.
Very nice overview, very informative. Questions from the audience? Yes, please.
Frank, Donald Bruce from Ethics. Presumably, Theranostics have, insofar as you've looked at them, certain particular features of a translation from something you've detected to a means of then determining doses and whatever else in a way that's different when they're separate. So have any particular features emerged as you have looked at these in practice?
Not that I know of, but I would assume that we will keep our minds open, and if such challenges arise, we are open for dialogue. We need to solve the problems together, and I think one of our colleagues mentioned that FDA is a problem. I don't know if FDA is a problem. I mean, FDA has lots of problems, just like we all do when developing new product and trying to enforce policy when it comes to regulation. But I think we need to also learn as a society to solve those problems in dialogue together. And it's certainly not a solution to wait until we have such problems that we approach our politicians, take the problem to Washington, D.C., and then wait for politicians to solve it. That usually doesn't work well, because as soon as the politicians get into the agency, which they don't quite understand how it works, we have a lot of nonsense going on. So I know I don't answer your question in a specific way, but I'm not aware of any specific challenges that we actually had to deal with.
Could I share an experience from a public point of view? The Engineering and Physical Science Research Council had a series of grand challenges in nanotechnology, which it would fund a big project, and it looked at nanomedicine for one of these. And I was on the Societal Issues panel of the Research Council at this time, and we said, okay, so what would be a good subject for a grand challenge in nanomedicine? And a range of different possibilities were looked at. And we decided to do a public consultation. And the one that the public picked out as a bit more iffy, a bit more dubious, was the idea of a sort of black box theranostic device that translated directly, as it were, from something you detected to something you then fixed. And people said, I don't like the idea of something where you're determining where it's kind of such an enclosed box that either my doctor or myself hasn't got any way of, in a sense, altering it if it doesn't work properly. There was just a public perception. I just sort of feed that into, that was as kind of an outsider's view that came quite strongly from that study.
Yes, fair enough. I take your comment also as a reminder to share with you, particularly in the US, I think in the past 3-4 years, we have a very strong interest in patient preferences, patient preference research, patient-reported outcomes, also in the context of product development and speed up of product development, because patient-reported outcomes and outcomes measures can be validated in the context of correlates or surrogates for clinical endpoints, which sometimes take a long time, and hopefully we can leverage that. But again, it's a question of doing the science right, validating such methods, keeping it transparent, and being considerate of the impact that has for society or any of the stakeholders.
So based on some of the things or thoughts we've seen before, right, you would expect that there is quite a lot of activity going on now in the field of nanomedicine to develop tools or technologies, whether they're theranostic or not, to figure out which patients to include in trials. Do you actually see that? Do you see that people are addressing this heterogeneity that there is in tumor accumulation?
Yes, patients at least look for, I mean, think of the opportunity to enrich trials for particular subsets in a, let's say, disease category. I think it's one of the leverages of precision, personalized, or whatever you call it, medicine, that would give you the tools, and then, of course, it right away goes into the question, so, yes, if you use biomarkers, for example, and if this is based on a specific technology, what are the evidence standards to validate a biomarker so you can actually use it? And this is also a dialogue. I mean, let's be honest, the agency alone cannot define biomarkers, and even though we are tasked to develop standards or evidentiary standards for biomarker development, it really depends what kind of disease it is. So one has to be specific enough to understand the challenges and the caveats, and one has to create a constructive dialogue between the stakeholders so that those issues can be addressed. And one shape, unfortunately, doesn't fit all. So there is not a general solution to all the problems, unfortunately, or maybe fortunately, because if it wouldn't be for the great diversity of human beings, medicine might just as well be a science and not an art.
No. Okay. Last question, Cassie.
Frank, thank you for making things a little bit more clearer. But if I understand well, I mean, the atmosphere of your lecture, in a way, what you're saying, Theranostics are drugs. We treat them as drugs, in a way.
Could be devices, could be biologics.
Yeah, depend. But depend what they are, then we treat them in the same way they are. And actually, from what I learned before from the FDA said, and nanodrugs is also a drug. I mean,
yes.
And we treat it as a drug. Okay, so first of all, we agree about this. Okay, now I have a question. If you go to details, which are very critical in your case, as you show, let's say we take a very simple parameters, which is very critical to the nanoparticles, or at least everyone believes is critical. This is the size distribution. You actually have a few highlights about this. Particle size distribution. Okay, now the FDA, although it requires a lot of information about this, but in a way it doesn't tell, since we know there are many, many methods, work on very different principles, I don't want to go into it now, and also can be expressed in a very different way, and they will come with a different answers that can be 20% different, even more sometimes. Now, we, the users, I mean, the people that apply for the FDA, actually, for us, it's a mystery what the FDA want at the end, in how they want to present the data. They don't want us to present it in 50 different ways, because and how we have to deal with such an issue.
Well, I guess what is the most meaningful method to characterize your product, right? I see my colleague Anil Patri working in. He's spent a whole career in the area of characterizing material. Of course, I think there has to be some consensus about some of the basics, right? And then there are probably some challenges to each and every product. Simple products are probably easier to characterize. And yes, there may be a spectrum of, sizes that make your product. As long as you can define it, as long as your process in your manufacturing is stable and reproducible, and you can show that, well, clinically, this is what I get with that, I think it's fair game. I don't think that FDA is holding back things just because we don't have the ability to ultra-fine characterize each and every element of a particular
It doesn't,
drug.
But it's just not clear enough how you want to get the data.
Well, since Anil is here, maybe I turf this to him. And Anil, what would you say?
We'll cover it at 5 o'clock.
It has to be turned up. Just continue speaking. They will turn it up.
Okay, so we can pick up the discussion after the the plenary talk at 5. Thanks again. Thank you again.